Hi everyone, my name is Dr. Aadil Sen. Today we are going to talk about biochemistry revision for Fmge students, and this revision is going to help you a lot. So let's begin. Phenylalanine, which is essential amino acid, when it is acted by phenylalanine hydroxylase enzyme, it gets converted into tyrosine, and the tyrosine is non-essential amino acid. Once this tyrosine is formed, from this tyrosine we get catecholamines, thyroid hormones, and melanin. These are the derivatives of tyrosine. Okay, catecholamines include epinephrine, norepinephrine, and dopamine. And the thyroid hormones are responsible for brain development. And the melanin is a pigment which gives color to our skin. See, what are the uses of tryptophan? From tryptophan, there is a formation of niacin, which is vitamin B3. Remember MCQ, that is from 60 milligram of tryptophan we get 1 milligram of niacin. From tryptophan we also get serotonin, which is also known as 5-hydroxytryptamine, and from this 5-hydroxytryptamine we get melatonin. Okay, remember in maize there is a protein which is known as zein protein. That zein protein lacks tryptophan. In exam you will get question that melanin is formed from tyrosine. Melatonin is formed from serotonin, but if serotonin is not in options and tryptophan is given, mark tryptophan. How you can remember? Tyrosine and melanin, they are small words. Melatonin and tryptophan, they are bigger words. So remember like this. Now let's see some diseases. See this, phenylketonuria. It is due to deficiency of phenylalanine hydroxylase. As you can see here, if this enzyme is deficient, this tyrosine will not be formed. Once this tyrosine is not there, these derivatives will not be formed. If T3 and T4 are not formed, the patient will be mentally retarded. It means there will be mental retardation, which is the new name of mental retardation, that is intellectual disability. And the patient will be having mousy or musty odor. Also remember, the patient will be having fair skin because the melanin will not be formed. Fair skin means white skin. There is a condition which is known as albinism, which is due to deficiency of tyrosinase. As you can see here, this tyrosine is here. Actually, once this tyrosine acted by tyrosinase enzyme, it gets converted into melanin. Okay, if tyrosinase is not there, melanin is not formed. Therefore, the patient will be having fair skin. And the patients of albinism, they are known as albinos. They are totally white. Their hair is white, eyebrows is, eyelashes, everything is white. Okay, let's see another very important disease, that is pellagra. It is vitamin B3 deficiency, that is niacin, and you will be having 6 Ds. Remember these 6 Ds: diarrhea, dermatitis, dementia. If these three things are not treated, the patient can die. And the fifth D is delirium, and sixth D is depression. And this dermatitis is in the fashion of Cassell's necklace. And this dermatitis you can see here over the neck is in the fashion of necklace, which is known as Cassell's necklace. And you can also see here the dermatitis over the dorsum of hand. There is another disease which is known as Hartman's disease. It is due to tryptophan deficiency. Once the tryptophan is not formed, we know niacin is formed from tryptophan. If tryptophan is deficient, niacin will not be there and the patient will be having pellagra like symptoms. So this was a complete topic. Let's see another topic which is very important, that what is the composition of creatine. One is creatinine, another is creatine. Let's see what is the composition of creatine. Remember a mnemonic, that is GAM. G for glycine, arginine, A for arginine, and M for methionine. It means there is a composition of 3 amino acids: glycine, arginine, and methionine. Let's see complete reaction. Once glycine and arginine combine in the kidneys by the help of an enzyme that is known as transaminase, there is the formation of goniodoacetate. And this goniodoacetate enters into the liver where it gets converted into creatine. Here is a group which is added to goniodoacetate which is known as SAM, S-adenosyl methionine. And this S-adenosyl methionine is the universal methyl donor. It is the universal methyl donor. SAM is the universal methyl donor which gives methyl group to the goniodoacetate, and there is a formation of creatine. Now this creatine is going to enter into the muscle where it gets converted into creatine phosphate, and the creatine phosphate is also known as phosphocreatine. Here the enzyme works, that is creatine kinase, and the kinase is uses ATP. And also remember that creatine phosphate or phosphocreatine is the immediate source of energy during exercise for 8 to 10 seconds. For 8 to 10 seconds. And from creatine phosphate we get creatinine. And finally this creatinine is excreted through the kidneys. Now this is complete. Now let's see another topic, that is gluconeogenesis. Gluconeogenesis. For gluconeogenesis, remember rule of 2, that it occurs in 2 compartments, in 2 organs, and in 2 situations. In 2 situations. Okay. In 2 compartments, that is mitochondria and cytoplasm. Remember, any pathway which occurs in 2 sites or in 2 compartments, that always starts from mitochondria. Like we will study further some pathways which occurs both in mitochondria as well as in cytoplasm, but every pathway which occurs in 2 compartments always starts from the mitochondria. And this gluconeogenesis occurs in liver mainly and in kidneys also. In 2 situations, fasting and starvation, and in diabetic patients. Now let's see what are the substrates of gluconeogenesis. Actually, what is gluconeogenesis? It is the formation of glucose from non-carbohydrate sources. What are the non-carbohydrate sources? You can see pyruvate, lactate, glycerol, propionic acid, glucogenic amino acids, glucogenic and ketogenic amino acids, and any intermediate of TCA cycle or any intermediate of Krebs cycle. TCA cycle is also known as Krebs cycle. Now, you know that acetyl CoA is not a substrate of gluconeogenesis. Why? Because acetyl CoA is not an intermediate of TCA cycle. You can see this note point that acetyl CoA is not an intermediate of TCA cycle. Therefore, It is not a substrate for gluconeogenesis. And this was the previous question for your FMG exam. Now let's see another important topic, that is glucose 6 phosphate dehydrogenase deficiency, or simply G6PD deficiency. Let's see some important points about this enzyme, that it is the rate limiting enzyme of hexose monophosphate pathway, and or it is the first enzyme of HMP pathway, or it is the first most common enzyme deficiency. And what is the second most common enzyme deficiency? That is pyruvate kinase deficiency, which is the enzyme of glycolysis. Remember another point, that is no ATP is formed from HMP pathway. It's a very important point. We don't get any ATP from the HMP pathway. There's a question that if any enzyme of HMP pathway or glycolysis is deficient, can result hemolytic anemia. How? How it leads to hemolytic anemia? If there is deficiency of any enzyme in HMP pathway or glycolysis. Let's see. We know that RBC only uses glucose in every state, whether the RBC is in the fat state, fasting state, or starvation, or any state you are. RBC always uses glucose. Okay. Let's see glycolysis enzyme deficient. If there is a glycolysis enzyme deficient, glycolysis will not occur. Therefore, ATP will not be formed. We know this pump uses ATP, and this pump will not work without ATP. Therefore, there will be damage to cell membrane, and finally the hemolysis will occur. That is the breakdown of RBCs. This is a case for hemolysis in RBCs due to glycolysis enzyme deficiency. Now let's see HMP pathway, where if there is enzyme deficiency and you will get the hemolysis. So let's see. For this, remember that there is oxidized glutathione and reduced glutathione. Okay. Once NADPH added to oxidized glutathione, it becomes reduced glutathione. Actually, this NADPH we get from the HMP pathway. If HMP pathway is occurring, we will get NADPH. Okay. Actually, from HMP pathway we get NADPH. Okay. And this NADPH helps to convert oxidized glutathione into reduced glutathione. And the enzyme responsible here is glutathione reductase. Okay. And this enzyme needs vitamin B2, which is very important. And this is the MCQ also. Once reduced glutathione is formed, now we know in RBCs, hydrogen peroxide, that is H2O2, keeps on accumulating, and we have to remove this waste material from the RBCs. How we can remove? Now this H2O2 will add to reduced glutathione, and it will be converted into water, and again we will get oxidized glutathione. But there is an enzyme that is known as glutathione peroxidase, and it is very important enzyme that it needs selenium to work. Okay. If selenium is not there, this enzyme, that is glutathione peroxidase, will suffer. It will not work. So our topic was that if there is any enzyme deficiency in the HMP pathway, we will get the hemolysis. But how it will occur? If HMP pathway not working due to any enzyme deficiency, that reduced glutathione is not formed. What I have told you, that from HMP pathway we get NADPH. Okay. If any enzyme is if any enzyme is deficient in the HMP pathway, we will not get NADPH. Once the NADPH is not formed, we will not get reduced glutathione. If reduced glutathione is not there, okay, this H2O2, that is hydrogen peroxide, will accumulate inside the RBC, that will lead to hemolysis. Okay, this is the case for HMP pathway enzyme deficiency. Now let's see another important topic, that is glycogen storage disease. Here I am going to give you an quick mnemonic, that is victorious people can always make history today. By this mnemonic, you can remember the type of glycogen storage disease. V for von Gierke's disease, P for Pompis, C for Cory's, A for Anderson's, M for McArdle's, H for Hers, and T for Tauris. Okay. Now, this von Gierke's disease is due to deficiency of glucose 6 phosphatase enzyme. For this von Gierke's disease, I want you to remember some important points, that patient will be having gout. Patient will be having gout, that is hyperuricemia. Hyperuricemia is the gout. And the patient will be having hyperlipidemia due to increased lipids. They will be deposited over the face, and therefore the patient will be having doll-like faces due to hyperlipidemia. And also the patient is going to have severe hypoglycemia. And these are the points which can be given in the exam, so be cautious about these points. And you will if you will see hyperuricemia, severe hypoglycemia, go for von Gierke's. In Pompey's, you will be having the deficiency of acid maltase. Actually, this enzyme is present in lysosomes, so therefore if this enzyme is absent, we will get the Pomp's disease. That's why this Pomp's disease is known as lysosomal storage disease. It's actually a lysosomal storage disease due to acid maltase deficiency. And in Pomp's disease, in exam you can get the image of a child who will be having some cardiac problems like cardiomegaly, in enlargement of the heart, and the tone of the muscles of a baby will decrease, and patient will be having hypotonia. These are the cases or points which can be given in the question. So remember these points, they will help you. Cory's, it is due to deficiency of debranching enzyme. A for Anderson's, due to branching enzyme deficiency. And in Anderson's, you can get the history of cirrhosis. McArdle's is due to deficiency of muscle glycogen phosphorylase. Hers is due to hepatic glycogen phosphorylase. Tauris is due to phosphofructokinase, which is the rate limiting enzyme of glycolysis. Phosphofructokinase. Let me tell you that how to remember these things, these diseases. Remember a mnemonic, that is A, B, C, D. So very easy. A for Anderson's, B for branching, C for Cory's, D for debranching. Okay. Also, this one is also easy. M for McArdle's, M for muscle glycogen phosphorylase, H for Hers, H for hepatic glycogen phosphorylase. Also remember that muscle glycogen phosphorylase is also known as myophosphorylase. Myophosphorylase. This was the Fmge exam question. Now let's see another important topic, that is Chargaff's rule. See this question. If content of adenine is 15%, what is the amount of guanine in DNA according to Chargaff's rule? Before doing this question, let's see some points that in DNA we have purine, pyrimidine. Purines include adenine and guanine, and the pyrimidine include thymine and cytosine. We know that this adenine makes a double bond with thymine, and the guanine makes triple bond with cytosine. See what is question saying, that adenine is 15%. Adenine is 15%. If adenine is 15%, thymine will be thymine also will be 15%. 15 plus 15 equals to 30. Okay. Now question is saying what is the amount of guanine? What is the amount of guanine? See, if total DNA equals to 100%, if we have 100% DNA, now it's given that adenine is 15%, therefore 10 adenine will bind with 10 thymine, that equals to 30. Now the guanine, that will be 35, and this cytosine also will be 35. 35 plus 35, that equals to 70. Here is it is 30%, and here it is 70%. Therefore it comes 30 plus 70, that equals to 100. Now what is the amount of guanine? The answer will be 35%. It is so easy to remember, no difficult mathematics here. Now let's see some pathways and site and their rate limiting enzyme. Can be asked in your exam, so remember these points very well. Glycolysis, it occurs in cytoplasm, and the rate limiting enzyme is phosphofructokinase. Krebs cycle, mitochondrial matrix, rate limiting enzyme is isocitrate dehydrogenase. Glycogenesis is the formation of glucose. Glycogenesis is the formation of glycogen. Glycogen is a storage form of glucose in the liver or in muscles. And the site is cytoplasm, mainly occurs in the liver and skeletal muscles. And the enzyme is glycogen synthase, very simple enzyme. Glycogenolysis, it is a breakdown of glycogen, and same it occurs in cytoplasm, mainly in liver and skeletal muscles. And the enzyme which is rate limiting enzyme, that is glycogen phosphorylase. Now let's see another important topic, that is glycolysis. Glycolysis is the breakdown of glucose. Okay. See, this glucose gets converted into glucose 6 phosphate, then glucose 6 phosphate gets converted into fructose 6 phosphate, and from fructose 6 phosphate we get fructose 1,6 bis phosphate, and this fructose 1,6 bis phosphate gets converted into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. I'm giving you some compounds which are very important to some substrates which are very important to remember here. Now this glyceraldehyde 3 phosphate undergo various reactions. Finally it gets converted into phosphoenol pyruvate. From phosphoenol pyruvate we get pyruvate. Okay. This is the case of glycolysis. The end product for glycolysis is the pyruvate. Now remember some important points, that you can see here that from pyruvate, I have told you that pyruvate is one of the substrate for gluconeogenesis, that from pyruvate we can get the glucose because it is the non-carbohydrate source. Okay, from pyruvate, how we can get the glucose back? You can see I have marked here the three red marks, three red arrows. These three red arrows show the irreversible reactions. Here are three irreversible reactions which are occurring. But what are the enzymes which are present here? In first red arrow, the enzyme will be hexokinase or glucokinase, and it will be responsible for irreversible step of glycolysis. And the second red arrow shows phosphofructokinase, and the third arrow shows pyruvate kinase. And what are the irreversible steps of gluconeogenesis? You can see the pyruvate gets converted into phosphoenol pyruvate by the enzyme of pyruvate carboxylase, which needs biotin. And here you can see another irreversible reaction. We have to reverse this, that is fructose 1,6 bisphosphate to fructose 6 phosphate. The enzyme here responsible for to convert this reaction, that is fructose 1,6 bisphosphatase, which is the rate limiting enzyme of gluconeogenesis. And another red arrow here which is irreversible, and it can be reversed by glucose 6 phosphatase, which is absent in skeletal muscles. And by this process you can get the glucose. And you can see the reversible enzymes or enzymes which are needed in the gluconeogenesis, that is glucose 6 phosphatase, fructose 1,6 bisphosphatase, and the pyruvate carboxylase. These are the three enzymes. Here you can see that biotin deficiency occurs due to intake of raw eggs. If you take raw eggs, you will be having biotin deficiency, because this avidin which is present in the raw eggs, it inhibits biotin. And the patients who are having biotin deficiency will be having alopecia, that is hair fall. We know that there are two types of glycolysis, that is aerobic and anaerobic. In aerobic we get 7 ATPs, and from anaerobic glycolysis we get only 2 ATPs. Now remember this chart, which is going to be very important, and every time you will get 2-3 questions from this chart itself. I have told you from tyrosine we get melanin, thyroid hormones, and catecholamines, and from tryptophan we get 5-hydroxytryptamine, melatonin, and vitamin B3. From arginine we get nitric oxide, and from histidine we get histamine, and where we get from what cells we get histamine, that is mast cells. Fmge exam question. And this glycine is also important for heme synthesis and creatine synthesis. Adenosine, once this is acted by adenosine deaminase, it gets converted into inosine. Now what is the importance of adenosine deaminase? If there is increased adenosine deaminase, it is highly suggestive of tuberculosis. Very important. If the ADA levels is decreased, it is suggestive for severe combined immunodeficiency, where you will see decreased ADA. In this SICKID, you will get both B and T lymphocytes affected. Now let's see another disease which is known as maple syrup urine disease, which is very important disease. repeatedly asked in the exam. You know, there are branch chain amino acids like valine, leucine, isoleucine. You can remember a mnemonic that is VIL, VIL or VLI. Okay, this branch chain amino acids is a exam question itself. What are the branch chain amino acids? They get converted into branch chain keto acids, and these branch chain keto acids gives their product. But here the enzyme works branched chain alpha keto acid decarboxylase or dehydrogenase. The new name is decarboxylase, and this enzyme actually is an enzyme complex and it needs vitamin B1. Okay, if this enzyme is deficient, actually this enzyme is deficient itself in this disease. If this enzyme is deficient, patient will be having mental retardation, because these branch chain acids, branch chain amino acids, actually these branch chain amino acids are very important for the brain. These are very important for the brain. Once these are not utilized, once these are not utilized, they can result mental retardation. Also remember that the patient will be having burnt sugar smell in the urine, which is the characteristic inside patients, and you will be having this this term, that is burnt sugar. This will be given in the question, which is very important. Let's see another topic, that is collagen. This collagen is rich in the glycine amino acid, very important. What is the primary structure of collagen? You will see the collagen will be having glycine, then another amino acid, then another amino acid. It means every third amino acid is glycine. And this XY, this X can be proline or hydroxyproline, and this Y can be lysine or hydroxylysine. See, this collagen synthesis, formation of collagen needs some enzymes and vitamin C. Actually, collagen synthesis enzymes, the enzymes which are responsible for the synthesis of collagen needs vitamin C. What are the enzymes? Prolyl hydroxylase and lysyl hydroxylase. These two enzymes need vitamin C, which is very important. See, Fe3 positive, which is a ferric form of iron, and Fe2 positive, which is a ferrous form of iron. How to remember? Ferrous. You can see S at the end of ferrous. So remember, Fe2 positive is the ferrous form of iron, and this is easily absorbed. How it is going to occur? That ferric form of iron gets converted into ferrous form of iron by the help of vitamin C, and the ferrous form of iron is easily absorbed. Once the patient will be having vitamin C deficiency, which is known as scurvy. So you can remember that if there is scurvy, in scurvy, what is going to happen? Vitamin C is not there. Once vitamin C is not there, the ferric form of iron is not going to convert into ferrous form of iron, and nothing will be absorbed. It means the patient will be having anemia. If iron is less, hemoglobin will be less, and the patient will be having anemia. Also remember, I have told you that here you have to remember that the wall of the blood vessels needs vitamin C, or you can say the wall of the blood vessel contains collagen, which is very important for the wall of the blood vessel. If the collagen is not there, or you can say if vitamin C is deficient, collagen will not be formed properly, because the enzymes of collagen are affected. That leads to bleeding gums, because the wall of the vessel becomes weak due to deficiency of vitamin C. Okay, and the patient will be having poor wound healing, therefore collagen is affected, because collagen is affected, and the patient will be having gum bleeding. If there is infants, if there are infants who are having scurvy, that's known as Barlow's disease. Also remember, I have told you that vitamin C helps in the iron absorption. Another condition which is known as fluorosis. There are two types of fluorosis. That one, the first one is dental fluorosis. Another one is skeletal fluorosis. Dental fluorosis occurs in less than 8 years of age, and skeletal fluorosis is due to increased systemic levels of fluoride. Okay, and the skeletal fluorosis is associated with uh, osteosclerosis. It is associated with osteosclerosis. What is osteosclerosis? It is the abnormal hardening of bone due to increased bone density. Increased bone density. As you can see here, increased bone density. Increased bone density, which is seen in the skeletal fluorosis. This time in Fmge exam, the question was asked. That was a case of fluorosis, but the question was asking that in fluorosis, which element is responsible? That is fluorine. Fluorine is responsible for the fluorosis. This is the case of dental fluorosis, and this is the skeletal fluorosis. Also remember some elements like macro elements and micro elements, which are also known as trace elements. In macro elements, you will get sodium, potassium, chloride, calcium, phosphorus, and magnesium. This question came in Fmge exam 2 times. So remember that calcium is the macro element, and in micro elements you are going to get iron, iodine, selenium, copper, chromium, manganese, fluoride, and zinc. Now let's see urea cycle, which is also known as ornithine cycle or Krebs-Henselt cycle. And the site of urea cycle is liver, mainly mitochondria and cytoplasm. It means I have told you this pathway is going to start from the mitochondria itself. Okay, and what is the rate limiting enzyme? That is CPS1, carbamoyl phosphate synthase 1. Exam question, and this is also the exam question. In exam they can give you both options, and suppose if A option is mitochondria, B option is cytoplasm, C option can be both, and mark this one. Nitrogenous waste is excreted from body in the form of urea. Okay, now let's see another important point, that this urea is having this structure, but in urea this carbon comes from where? It comes from carbon dioxide, and this nitrogen they come from NH3 and aspartate. It means one nitrogen is given by aspartate and another nitrogen is given by ammonia. Let's see another topic, stop codons. What are the stop codons? UAA, UAG, UGA, UAA, UAG, UGA. UGA, it codes for 21st amino acid, which is known as selenocysteine. UAG, it codes for 22nd amino acid, which is known as pyrrolysine. In start codons you have to remember AUG. This AUG in eukaryotes is responsible for the formation of methionine. It means AUG codes for methionine, and in prokaryotes it codes for formyl methionine. Okay, now let's see another important topic, that is inhibition. First one is competitive inhibition, non-competitive inhibition, uncompetitive inhibition. I don't want you to teach this detail, but I want you to remember the values of Km and Vmax. In competitive inhibition, you will see the Km is going to increase, and in non-competitive inhibition, Vmax is going to decrease. You have to make the cross like this so that you can remember, and these two things will be same. Make a cross and write increase decrease same, and in uncompetitive both decrease. Let's see another point, that fluorocitrate, it is a competitive inhibitor of aconitase. Aconitase is the enzyme of TCA cycle, and the malonate is the competitive inhibitor of succinate dehydrogenase. All oxidases require cofactor copper except xanthine oxidase and sulfite oxidase. They need molybdenum. Very important. Remember for xanthine oxidase. See guys, that ammonia is detoxified in brain or major tissues by glutamine, and the ammonia is detoxified in muscles by alanine. In muscles, ammonia is detoxified by alanine. Ammonia is detoxified in brain by or major tissues by glutamine. A very important cycle which is known as Cahill cycle or glucose alanine cycle. See, in the fasting muscle, if the muscle is fasting, you can see in the glucose which is present inside the muscle is going to convert into the pyruvate, and this pyruvate is added by amino group and gets converted into alanine, and this alanine from the muscle is going to enter into the bloodstream, and from blood it will go into the liver, and in liver again the amino group will be removed, and there will be formation of pyruvate, and from pyruvate we will again get glucose through the gluconeogenesis process, and again this glucose will enter into the blood vessel, and then it will enter into the muscle, and this cycle will continue in the fasting muscle. Therefore the KL cycle occurs in the fasting muscle. See, alpha ketoglutarate, which is a substrate of TCA cycle, and once the NH2 is added to alpha ketoglutarate, it becomes glutamate, and once amino group is added to glutamate, it gets converted into glutamine, and the enzyme here is glutamine synthase, and therefore ATP is used here. Remember, it is actually synthetase, glutamine synthetase. Actually synthetases use ATP, but synthase does not use ATP. Okay, see guys, that methionine we take in the diet because it is essential amino acid. Remember, we are going to discuss some important points about homocysteine and cysteine as well. If the patient is having excessive homocysteine levels in the blood, what is going to happen? is associated with atherosclerosis. Very important, can be asked in the exam. Suppose in question they are giving the history of stroke or MI or pulmonary embolism, and if in option if they give excessive homocysteine levels in the blood, you can mark that. And you will be given a young guy who is having stroke or MI or pulmonary embolism, go for excessive homocysteine levels. See guys, homocysteine is having SH group, that is sulfhydryl group. Okay, in excessive homocysteine or homocystinuria, you will be having excessive homocysteine in the blood. Another point is homocysteine. This homocysteine is made up of two homocysteines. It is made up of two homocysteines. It means it will be having two sulfhydryl groups, that is SH and SH. Okay, but once the homocysteine is formed, what is going to happen? This SH and SH will combine, therefore, and the two hydrogens combine, and two hydrogens are removed in the form of water. Okay, and there will be the bond like this. The bond between sulfur and sulfur is a disulfide bond. Okay, so remember, in cysteine there is one sulfhydryl group, in cysteine there are two sulfhydryl groups. And this excessive homocysteine levels in the blood is associated with homocystinuria. It means the homocysteine will come into the urine. It is homocysteine. Okay, there is a receptor which is known as adenosine receptor. Its stability is because of extensive disulfide bonds between. Its stability is due to extensive disulfide bonds between. I have told you that disulfide bonds are present in what? It is between two homocysteine. It is between two homocysteines. When two homocysteines combine, they form homocysteine. Okay, so remember, when two cysteines combine, they form cysteine. Therefore, the bond will be between two cysteines so that they can form cysteine. Remember that what are the PCR steps? The mnemonic is DAE, die. The first one is denaturation, which occurs at 95 degrees Celsius. Then aniline, where we have to add primer. And the third step is extension, where the polymerization of the DNA occurs. In PSR, what we do is actually we made the multiple copies of DNA. See guys, that what is the marker of B1 deficiency, thymine deficiency, that is known as RBC transketolase activity. We have to check RBC transketolase activity because this enzyme needs vitamin B1. And what is the marker of B2 deficiency? That is RBC glutathione reductase activity. Now let's see two diseases which are very important for every exam, that is Wilson's disease and Menkes hair disease, or Menkes disease simply. Wilson's disease is autosomal recessive. It is due to defect in ATP7B gene. In Wilson's disease, you will get excessive copper, and the copper is deposited in liver. Once the copper is deposited in liver, it leads to cirrhosis. Once the copper is deposited in basal ganglia, it leads to wing beating tremors. Also remember that copper is also deposited in the descendants membrane of cornea, which leads to Kasher-Fleischer ring. As you can see here, the copper is deposited in the descendants membrane of cornea in the form of a ring which is known as KF ring or Kaiser-Fleischer ring. Same image you can see here. This was the image which was given in the Fmge Jan exam. This is the KF ring. Also remember, in Wilson's disease you will get the less ceruloplasmin. Also remember that Wilson's disease is also known as hepatolenticular degeneration. Why? Because the liver is involved, that is hepatolenticular. It means lentiform nucleus is involved. There is degeneration of lentiform nucleus. Lentiform nucleus itself is a part of basal ganglia. That's why you will get wing beating tremors. If lentiform nucleus is involved, also this patient is associated with Parkinsonism. And for diagnosis, we are doing some tests. The most specific test is increased urinary copper excretion, and the gold standard test is liver biopsy. And for treatment, we are giving some chelating agents like penicillamine, trientine, or zinc. Now let's see Menkes disease. It is X-linked recessive due to ATP7A defect, ATP7A gene defect. It is due to copper deficiency. Wilson's disease copper excess, but Menkes disease is copper deficiency. Once the copper is deficient, copper is needed for some enzymes like lysyl oxidase, tyrosinase, both will be affected. Once the lysyl oxidase is affected, the patient will be having brittle or kinky hair because the collagen is affected. Actually, copper also needed for collagen. Once the copper is not there, collagen will be affected and the patient will be having brittle or kinky hair. And this tyrosine is also affected, therefore the patient will be having gray hair. Also remember, in Wilson's disease you will be having sunflower cataract. Sunflower cataract. Also remember, in both Wilson's disease and Menkes disease, the ceruloplasmin will be decreased. Let's see another important condition. You have to remember this enzyme, that is hypoxanthine guanine phosphoribosyl transferase, that is AGPRT. If this enzyme is completely deficient, complete deficiency, the disease is known as Lesch-Nyhan syndrome. If there is partial deficiency of this enzyme, it leads to Kelly-Sigmiller syndrome. In Lesch-Nyhan syndrome, the patient will be having hyperuricemia, that is gout, and the patient will be having self-mutilation behavior. And in partial deficiency, you will be having hyperuricemia, that is gout, but the patient will not be having neurological symptoms like self-mutilation behavior. You can see this lip biting, nail biting. Okay, this is a case of Lesch-Nyhan syndrome, and this image can be given in the exam. Mostly this question is asked. Let's see some GAGs, glycosaminoglycans, which include keratin sulfate, dermatan sulfate, chondroitin sulfate, and hyaluronic acid. Hyaluronic acid. Okay, you have to remember that Keratin sulfate and chondroitin sulfate both are present in the cornea, but keratin sulfate is abundant in the cornea, or it you can say it is more than chondroitin sulfate. This was the neat PG question itself. This hyaluronic acid, H for hyaluronic acid and H for healing. It helps in the wound healing, and it is the longest gag. Hyaluronic acid is longest gag and is the only gag not having sulfate, and present in vitreous humor and synovial fluid, and it has role in cell migration during morphogenesis. When the structures of baby are formed, during that period the cell migration occurs, and it has the role in that. Also remember very high yielding image, that is acrodermatitis enteropathica. It is due to deficiency of zinc. Actually, it's not due to deficiency, exactly not due to deficiency of zinc. It is actually due to decreased absorption of zinc. And you will see the dermatitis over the peripheral parts, like over hands, over the genital area, over the face. Okay, enteropathica, it means the intestines are involved. Enteropathica, pathology in the intestines. Okay, let's see another point, that zinc is zinc in diarrhea. What is the importance of zinc in diarrhea? Why we give zinc in diarrhea? It reduces duration and severity of diarrhea. Yes, it is correct. Enhances immune response. Yes. Regulate absorption of water and electrolytes is also correct. Not synthesize it in body. Yes, we have to supplement it. And cofactor for carbonic anhydrase enzyme. It means all of the following are correct. Now let's see fatty acid synthesis. What is the first enzyme of fatty acid synthesis? That is acetyl CoA carboxylase. Second enzyme of fatty acid synthesis is fatty acid synthase, which require NADPH. It means in fatty acid synthesis we need NADPH, which is the exam question. All are dehydrogenases except. All are cofactors for dehydrogenase except. The answer is lipoic acid. Let's see some detail about dehydrogenases. These dehydrogenases remove hydrogen. It's not much clear. Let's see. These hydrogenases remove hydrogen called as oxidation, and this hydrogen which is removed by dehydrogenase, it is taken by following things. It's taken up by, it can be taken by NAD. It can be taken by NAD. Once it is taken by NAD, it becomes NADH. Okay, if this hydrogen is taken by FAD, it becomes FADH. If it is taken by NADP, it becomes NADPH. Okay, remember FAD is also known as vitamin B2, and NAD is also known as vitamin B3. This NADH, once it goes to electron transport chain, it gives us 2.5 ATP. FADH gives us 1.5 ATP. NADH gives 2.5 and FADH gives 1.5 in the electron transport chain. But I have told you in HMP pathway we get NADPH, but we don't get any ATP from the HMP pathway or from NADPH. No ATP is formed from the NADPH. And this NADPH used in the anabolic pathways like in the fatty acid synthesis, as I have mentioned above. Therefore, I have told you all are cofactors for dehydrogenase. These all are cofactors for dehydrogenase, but the lipoic acid is not the cofactor for dehydrogenase. But you can see lipoic acid is cofactor for pyruvate. It's a cofactor for pyruvate dehydrogenase in the which is actually the link reaction. Okay, pyruvate, once the pyruvate is formed, it gets converted into acetyl CoA. The enzyme which works here is the pyruvate dehydrogenase, and this is called a link reaction. Link reaction is simple. It is the conversion of pyruvate into acetyl CoA, and the enzyme is pyruvate dehydrogenase. And the uh, and the cofactor which works here, that is the lipoic acid. Cholesterol, fatty acids, triglycerides, ketone bodies are synthesized from acetyl CoA. What is the DASH diet? Dietary approach to stop hypertension. It is the approach to stop the hypertension by the help of diet. You have to, in diet, it contains high calcium, high potassium, high magnesium. Increase the number of fruits and vegetables, but decrease the sodium. We know that sodium increases water inside the blood, therefore it increases the risk of hypertension. So we have to decrease the sodium in the diet, and this was a question in the exam. Now let's see some repair mechanism and defect. The mismatch repair is seen in the HNPCC, hereditary non-polyposis colorectal cancer, and nucleotide excision repair can be seen in nucleotide excision repair defect is seen in xeroderma pigmentosa, and mismatch repair defect is seen in hereditary non-polyposis colorectal cancer. Homologous repair defect is seen in Bloom syndrome or Werner syndrome. Remember that xeroderma pigmentosa is autosomal dominant, and you will be the patient will be having these freckles. You can see these are the lesions over the face, which is seen in the xeroderma pigmentosa, and these lesions will be most likely over the face because it is more seen over the sun exposed areas, and it is associated with squamous cell carcinoma. Very important, and don't forget to remember this image. What are the lipotropic factors? Lipotropic factors mobilize triglycerides from liver cells to periphery, therefore prevent fatty liver. Actually, lipotropic factors are the factors which take the triglycerides from the liver cells to the periphery, therefore they can prevent the fatty liver. Example are polyunsaturated fatty acids, choline, folic acid, that's B9, B12 and methionine. Now see the question. In fatty liver, which is not given? We can give choline, B9, B12 because they are what? They are lipotropic factors, but we don't give ethanol. Ethanol is an alcohol which can itself lead to fatty liver. Therefore, it is avoided in the fatty liver. Also remember that there is an enzyme which is known as superoxide dismutase. Superoxide dismutase, SOD. One is mitochondrial SOD, second is cytoplasmic SOD. Mitochondrial SOD needs a cofactor, that is manganese, and the cytoplasmic SOD needs copper as a cofactor. So remember, M for mitochondrial, M for manganese, C for copper, and C for cytoplasmic SOD. Hope you have enjoyed this revision session, and in the next session we'll take up the electron transport chain and we will complete the entire biochemistry. Hope you have enjoyed this video. Thank you.